Well, welcome back from the break. In reality, as I record this, it's actually another day. You may have noticed from the background that, and in fact, I'm wearing a different shirt, that I am in fact recording this on a different day, and that's because of the amount of material that needs to be covered. Fortunately, the second half is going to be a little bit shorter, although we do run up against the clade of all clades, the largest clade of any terrestrial animals, and that is the insects. Once a class known as Insecta, then a subphylum known as Hexapoda, and I've referenced it as such in the presentation, but I understand there is still a lot of discussion about exactly what to refer to insects as a group. The current phylogenetics places the insects along with crustaceans into a single clade called Pancrustacea, which we'll get into when we get to that point in the presentation. But exactly where in the biological hierarchy this broad group of organisms that we refer to loosely as insects belongs is still subject to a lot of discussion. But we'll get there. First, let's look at the concept check question from last time and answer this question. So earthworms are often described as a tube in a tube. Explain what is meant by this description. Well, the first tube is really obvious. That's the outer body, what you might refer to as the body wall, which is made up of the epidermis plus the two layers of muscle tissue that surround the coelom. So there is your first hollow tube. The second hollow tube is the digestive tract suspended within the coelom. Remember, a coelom is a true body cavity, and the digestive tract of the earthworm is suspended in it. And it is also a tube with the mouth at one end, the anus at the other end, and a cylinder, a hollow cylinder connecting the two openings together. So there is the basis for this description of a tube within a tube. The inner tube is the digestive tract. The outer tube is the body wall of the animal itself. And in the intervening space, you find the body cavity, the coelom. So you have the inner tube suspended in the cavity, the hollow cavity of the outer tube. That's how we arrive at the tube within a tube description. So let's move on and look at the other, or the second large group of Bilateria, and this is the second clade derived from the splitting up of the protostomia. And these are the ecdysozoans. And remember, the unifying physical characteristic is the presence of an outer coating, a body coating, an exoskeleton or a cuticle that is shed periodically through this process known as ecdysis. And we are only going to focus on two of the phyla within the ecdysozoans, but you may recall from earlier in the presentation when we looked at the diversity of the animal kingdom that there are a significant number of clades that do belong within this large clade, the ecdysozoans. We're just going to look at the nematodes and the arthropods as the two major clades within the ecdysozoan group. So let's begin with the nematodes. So nematodes are classically referred to as roundworms. This is because their body lacks any evidence of segmentation. They do have a body cavity, but it is a hemocoel or a pseudocoelom using the old classification. So that means it has a single layer of mesoderm-derived muscle tissue associated with it. It lines the outer rim of the body cavity, so it lies just under the body wall and the epidermis. So if we're moving from the outside to the inside, you have the cuticle, the epidermis, and then lying immediately under the epidermis is this single layer of longitudinal muscle that runs anterior to posterior. And that means that nematode worms don't burrow because they can't shorten and lengthen their body as an earthworm might do. What they can do, however, is flick their body from side to side. So they typically wriggle through their environment. Not as elegant as the movement of a worm, but it gets the job done nonetheless. And that interior body cavity, the pseudocoelom or hemocoel, well, that substitutes for a circulatory system to distribute nutrients that are absorbed from the digestive tract, and also to distribute gases through gas exchange. Gas exchange is primarily through diffusion through the skin of the animal, but to move it throughout the whole body and also to move it into the inner organ systems. We use the fluid. So it would, of the body cavity, it would be technically referred to as hemolymph. If we were to assign a name to it, we definitely would not call it blood. So let's take a look at I think I've got a picture of a nematode. No, I don't. So we're just going to have to recall the picture that you saw in lab. So if you cut through the cuticle of a nematode worm, what you find once you've dissected it open is a digestive tract, the reproductive organs. And remember that in nematodes, there are males and females. These are dioecious, a separate male and female organism. And you have the reproductive organs and the digestive tract suspended in the body cavity. They are not attached other than at the very ends of the organs to the inner wall of the nematode. They are in fact suspended in the fluid. And that allows the worm to flick its body in a whip-like fashion without damaging the organs, because they are suspended in the fluid, and that protects them from damage. If they were attached to the inside of the wall as the worm vigorously flicked itself from side to side, there would be very real potential for damaging the organs. And you may recall too that a key way of identifying the males from the females, apart from size, the females are always bigger, is the fact that the male has what I refer to as a hooked tail. So here you're looking at one is probably a male, because you can see that the tail here, let me just make sure the pen is working. The tail has this hook associated with it. That is very characteristic of the male nematode worms. And the reason why this is hooked is to allow the worm to deposit, the male worm to deposit the sperm into the receptacle, which is an opening in the outer covering of the female, so that he can actually attach to that receptacle and deposit his sperm there for the purpose of fertilizing the eggs. So the males always have this hooked tail, which is very, very characteristic. It's a very clear indicator of the male versus the female. Now, this particular worm is Ascaris. That is the nematode that you saw in lab as the dissection. What is quite remarkable about Ascaris is that it is in fact a parasitic worm, including a parasite of humans, and these worms can get up to 6 to 8 inches in length. Fortunately, it's not the adult worm that is encysted in human bodies. It's actually the juveniles, the larval stages. They tend to encysted in the muscles, and it can be extremely painful because of the presence of these cysts inside your muscles. How do you get infected? By eating poorly cooked animal products, particularly meat. Because if you eat meat that has the cysts in it, and you don't cook it properly, you don't kill the larvae, they will then hatch out in your body. The males and the females will then mate and release fertilized eggs, which will develop into the larval stage, which will migrate through your bloodstream from the digestive tract and encysted themselves in your muscles. And again, it is incredibly painful. So this is something that while you may think it's not something that you need to be aware of, with the advent of the free range movement and the increasing use of the free range habit of raising particularly pigs, there is the very real potential of those pigs picking up a nematode infection as they're rooting around in the soil. And if these are not carefully monitored, if the condition of the pigs is not carefully monitored, if it's not caught that these cysts are in the meat and you happen to purchase some of this and don't cook it properly, then you run the very real potential of getting infected. While the pigs were being raised in a sterile barn-like arrangement, that reduced the chances of picking up a nematode. So really it's 6 of one, half dozen of another. If you want to maintain the overall health of the pigs and reduce the possibility of, for instance, nematode and other parasite infections, then raising them in a large concrete floored pigsty in an enclosed building is the way to go. But if you want to maintain the physical and mental condition of the pigs and raise them in conditions that are more humane, then you would raise them outside and let them roam free. But then you run the risk of infections. It's 6 of one, half dozen of the other, pretty much. Most nematodes in the environment are usually scavengers or predators of small microorganisms, but some species of nematodes are parasites, both of animals and also of plants. Another major nematode parasite of humans is Trichinella spiralis, which causes trichinosis, again, from eating undercooked pork. And again, for the reasons that I've just explained, this has the probability of becoming more, or potentially more common, because again of this desire to raise pigs in more humane conditions as free-range animals. In the case of parasites of plants, if you were to attempt to grow tomatoes in the soil here in Florida, one of the things that you need to be aware of is the fact that if you take a bucket full of typical Florida soil, or if you even just take a handful of typical Florida soil, there are more nematodes in that handful of soil than there are, I believe this number is correct, than the total number of people in Florida currently. There are huge numbers of little parasite nematode worms in Florida soil. Previously, Florida agriculture got away with doing this by sterilizing the soil at the end of every growing season. And you would typically see wherever tomatoes and other plant crops were being grown in Florida, at the end of the growing season, large pieces of black plastic, huge spreads of black plastic were laid out over the ground. The edges were weighed down with soil to seal them down against the soil. And then you would see an individual in what looks like a simple spacesuit, because that's essentially what it is. It's a completely sealed hazmat suit, who would insert a hose under the plastic connected to a gas tank. That gas tank contained a gas called methyl bromide. Methyl bromide is a contact killer, and it kills everything. And it was perfect if you wanted to completely sterilize the soil and kill out all the nematodes and anything else that might be in there that would put your agricultural industry at risk. The problem was it did not discriminate. It killed all the things that you wanted to kill, but it killed everything that you wanted to retain in the soil too that might be friendly towards your agriculture. It was a complete soil sterilant, and it would sterilize the soil down to a depth of around 3 to 4 feet. Now, I used to work for the University of Florida at one of their research stations out in the Winter Garden area, the Winter Haven area. And while I was working out there, so this would be, it'd be close to 30 years ago now, the state announced that in a few years, methyl bromide would be banned completely and it could never be used again as a soil sterilant. And for a short time, that created a lot of angst amongst the growers in Florida, the agriculturalists in Florida. But eventually, it was realized that you could continue to grow things in Florida so long as you didn't grow them in the Florida soil. And that's when the switch to greenhouse growth came in. And that's why most of the winter tomatoes that you can buy now that are grown here in Florida are actually greenhouse grown, and why a large proportion of Florida agriculture now relies on greenhouse growth, because you can't sterilize the soil with methyl bromide anymore to get rid of the nematodes. They are a huge problem. Just to let you know how significant this is, if you grow the tomatoes in Florida soil, the first year you'll get away with it because you don't attract enough nematodes to be really lethal to your tomatoes. But if you grow tomatoes in the soil that second year without any sterilization, chances are really good that your crop will fail very, very early, because the level of nematodes in the soil now has increased because of your previous growth of the tomatoes. And this just increases the likelihood that those parasitic nematodes are going to destroy your next season's crop of tomatoes. They destroy the plants. So that's the reason why we do greenhouse growth these days. Or if you want to grow tomatoes in Florida, grow them in a pot. That way you can regulate and isolate your plants from the soil. Then another little nematode worm, and we've encountered this worm before back in chapter 47, is Caenorhabditis elegans, otherwise known as C. elegans, a little worm that contains around 956 to 959 total cells. I've seen both numbers. And every individual worm contains that exact number of cells. And this has been a really good model genetics organism. Why? Because a lot of the systems that are in genetic systems that are in play in C. elegans are also genetic systems that are in play in most terrestrial vertebrates, including humans. Surprisingly enough, once we had C. elegans genome sequence and we compared it against the human genome sequence, it turns out that we both have approximately the same number of genes, somewhere around 20 to 23,000 genes. So that just reinforces again an idea that we encountered in chapter 47, that the total number of genes is not the measure of complexity. It's what you do with the genes that determines the level of complexity. C. elegans has a limited number of Hox genes. It has the basic 7 to 8 cassette set of Hox genes. Humans, on the other hand, and all terrestrial vertebrates that possess jaws, which is all of them, have 4 cassettes of Hox genes. A lot more can be done with that greater number of Hox genes than you can do with just a single set of Hox genes. So again, it comes down to this idea of not how many genes you possess, but what you do with the genes that you do possess. So this is just a photograph of the juveniles encysted in muscle. This is fortunately pig muscle, not human muscle, but you can see the juveniles here encysted. It's the cysts present in the muscles that cause the muscle pain associated with trichinosis. So now we come up with we come up against the clade, the phylum, that is going to occupy a significant part of this presentation, or this part of the presentation, because we'll talk about the origin of arthropods, we'll talk about some of the clades of arthropods, but then we will address insects in some detail. And that's going to take up a large part of this section of the presentation. So the note here says 2 out of every 3 known species of animals are arthropods. You may recall what I said at the beginning of this chapter, when I started this presentation, that while we may have identified about 3.5 million species of organisms here on the planet, about 1 million of those that are known to science are insects alone, so not arthropods, but just a subset of arthropods, insects. And on the basis of what we know about the diversity of insects, we suspect that there are probably 3 to 4 million species of insects in total on the planet. We will never know the exact total because unfortunately many insects go extinct before we can identify them and describe them. And there are constantly new species of insects emerging as the Earth is constantly changing. So we'll probably never get a handle on the total number of insects present. That doesn't mean that we still can't estimate how many are there. And then we have to add in on top of that the fact that there are other arthropods. You have to think in terms of spiders, scorpions, ticks and mites. They form the Chelicerae, which is considered to be one of the more basal groups of arthropods. Then you also have in there the crustaceans, which are now considered to be part of a single clade with the insects, as I've already described on several occasions. We will get there. And then there are some related arthropods that are related to insects. They're considered to be basal to insects or basal to the pancrustacea, of which the insects are considered to be a group, or maybe are part of pancrustacea but are basal to the subgroup that insects belong to, whatever it turns out that they will be described as. I've used the term hexapoda because that's the subphylum that has been most recently assigned to insects. So there are a broad diversity of arthropods. When we start looking at the origin of arthropods, we start looking at the Cambrian period and the period of the explosion, when we first start to see organisms that have the basic layout of the arthropod body plan. That is an exoskeleton that is encasing a segmented body. On each segment, there's usually a pair of appendages. They may or may not be specialized for some purpose. Typically, in the more primitive arthropods, they were either used for locomotion or for protection. And maybe in some cases, you had both. You had appendages for locomotion and appendages for protection. One of the very, very earliest of all arthropods to turn up in the fossil record are these. You probably recognize these. This is a trilobite. So let me just put a note here. This is a trilobite. And what is characteristic of trilobites is over their long history in the fossil record, they show very, very little change in the basic body plan, and they show very, very little specialization. So when you're looking at this region, which is the abdomen, this does not change much. So trilobites as a group actually have a relatively long history in the fossil record. But the abdomen, which is the most highly segmented part of the body, doesn't show a great degree of specialization. Where you do see some specialization is in this area. This is called the cephalothorax. And so the reason why it's referred to as a cephalothorax, and you see this also in a number of other arthropods, instead of being covered by separate parts of the exoskeleton, the head and the thorax are covered by a single plate that covers both of them, and that's where we get the term cephalothorax from, this combination of the head and the thorax. So these are the trilobites. These are some of the very, very earliest of the arthropods to appear in the fossil record, and they have a long history in the fossil record, but show very, very little change just within the cephalothorax, if there is any change from over the history. One of the things that we do see as a function of arthropod evolution, and you do see this even reflected in the extant species today. So those species that are considered to be more basal to the arthropod phylogeny have many more segments and have a lot more appendages associated with those segments. And we've actually seen this before in a previous chapter where we did a comparison between Drosophila and the little brine shrimp. And the brine shrimp have many segments, particularly in the abdominal region, and those segments have little legs attached to them. Whereas when you look at the fruit fly, Drosophila, which is a more recent arthropod, being part of insects, it has a limited number of segments and only has 6 legs, 3 pairs that all insects have. And so what we see over the course of the evolution of arthropods is a decrease in the amount of segmentation, and also along with that, a decrease in the number of appendages, because each segment only has 2 appendages on it. But in the case of the later appearing arthropods, those appendices or appendices become vastly more specialized. So when you look at things like crabs, lobsters, what are referred to as the decapods, the marine crustaceans, you see that there are a large number of very highly specialized appendages. So when you look at the mouth parts, you look at the antennae, you look at the walking legs. In the case of the lobsters, you also look at the swimmerets, swimming legs. You see that they are modified for a number of different roles. You even look at the tails of the lobster, and you've got the telsons and the uropods, and those are themselves appendages that are for swimming purposes by flapping the tail up and down. So you do see an increase in the degree of specialization, along with the decrease in the number of appendages as a consequence of the decrease in the number of segmentation. This, of course, is likely to be a function of the Hox genes, because remember, the Hox genes are what lay out the body plan and establish the body plan. And one of the hypotheses that have been floated about the role of the Hox genes in the origin of the arthropod body plan is to look at 2 genes known as UBX, which we've talked about before. That's ultrabithorax. That's the gene which in the brine shrimp promotes the production of legs, hence the large number of legs. But in later arthropods, its role has been reversed, and UBX now becomes a suppressor of legs. And so in something like the Drosophila, the expression of UBX in the abdomen is actually suppressing the appearance of legs, whereas in the brine shrimp, the expression of UBX actually promotes the expression of legs. An associated gene called ABDA, or abdominal A, also influences the bithorax development. So ultrabithorax and abdominal A as Hox genes are both involved in the development of the bithorax structure, which involves the thorax and the abdomen, depending on which, depending on which group of arthropods you're looking at, you're either going to have a more highly developed abdomen with lots of legs, or you're going to have a relatively undeveloped abdomen in the absence of a large number of appendices. And this appears to be under the control of both UBX and abdominal A working together. A sister clade to the arthropods is the onychophorans, or the velvet worms. We've talked about these before earlier in the presentation. And the onychophoran body plan is basically very simple compared to arthropod. It lacks segmentation. It is a more solid body plan. There are appendages that represent a source of apparent segmentation, but the degree of segmentation in the body plan of onychophorans is far more simple than the degree of segmentation in arthropods. And so the question has been asked, what is the nature of the Hox genes in onychophorans that lack the degree of segmentation that we do see in arthropods? And is this a reflection of the Hox genes that are involved in the development of segmentation in arthropods? Are those genes present in arthropods and not present in onychophorans? And so UBX and abdominal A have been looked at because they are major drivers of segmentation in arthropods. Turns out that both of those genes are present in onychophorans as well. In fact, all of the genes that are responsible for developing the arthropod body plan, the Hox genes that are responsible for developing the arthropod body plan, are present in onychophorans as well. So what that says is that the evolution of the arthropod's highly segmented body plan with specialized appendages on each of the segments is not a function of the specific Hox genes that are present. It probably has something to do with either a change in the way those Hox genes are expressed or the timing of the expression of those Hox genes. And if you're not sure what I'm referring to there, you may want to go back and take a look at chapter 47 again and look at the section that covers heterochrony and differential growth, because that's where we talk about the development of the body plan and the differential development of different parts of the body on the basis of timing and location of expression. So that seems to be what may be at the heart of the origin of the arthropod body plan. Irrespective of what gave rise to it, it is the arthropod body plan, the basic nature of the arthropod body plan, that has made them so successful as both marine and freshwater animals, but also, and probably most spectacularly as far as we are concerned, as terrestrial colonizers and terrestrial animals. So what are the general characteristics? Well, these you should be pretty much familiar with. There is a lot of segmentation. Each of the segments has appendages, and those, a pair of appendages is the typical way we describe that. And those appendages can, and most frequently are, modified for very highly specialized purposes. So particularly when we start looking at the anterior end, we find the feeding appendages. These include both the appendages that move food to the mouth, but also appendages that are specialized to form the jaw structure. So even the jaws of arthropods are the product of modified appendages. We see sensory reception in the form of the antennae, again, modified appendages. If we're talking about the pincer claws, which can also be referred to as chelicerae, those are highly modified appendages as well. Then, of course, we know that all arthropods have legs. In fact, that's where the name arthropod comes from. Arthropoda means jointed leg, and it's a pretty fair description of what we've got. We've got a leg that is covered by the exoskeleton, but is divided up into segments by joints. And those joints contain a flexible material that links the rigid parts of the exoskeleton together to provide a flexible covering that allows the leg to bend. On the inside of the exoskeleton, muscles are attached to allow the appendages to move around these joints. It's one of the major reasons why arthropods are so successful, because not only do they have a very rigid, very tough exoskeleton that protects their body, it is broken up into segments, and those segments are joined together by waterproof connections that maintain the integrity of the covering, but now allow the exoskeleton to flex via the joints and the muscles that are attached to the inside. Then all arthropods, almost without exception, show a very high degree of cephalization. Being as mobile as they are, that means that they can move around in their environment. So it stands to reason that you want to move most of your sensory apparatus to that part of the body that is going to encounter the new parts of the environment first. That would be the anterior end. So you have your defensive mechanism, you have your feeding mechanism, you have your sensory mechanism in many forms, both touch and vision and taste, all located in the anterior end in the head. You have legs for mobility, then you have the abdomen that is itself also somewhat segmented, more or less depending on the species that you've got, but it is still obviously segmented. So the exoskeleton being made out of chitin is very rigid, but it also is a drawback for insects because chitin is digestible. So those organisms, those animals, predators that are able to hunt down and catch arthropods do have the ability to digest the exoskeleton and access the very protein-rich material of the body of the arthropod inside the chitin, but it does mean that you also need to have an apparatus to break it down. So that provides a layer of protection for the arthropods, but it also represents a major drawback, because if the animal wants to get bigger, the exoskeleton being rigid and somewhat inflexible prevents that unless the animal can shed it, hence ecdysis. So periodically, all arthropods have to shed their exoskeleton and secrete a new one. And during this period where they've shed their old skeleton and they're waiting for the new one to harden up, they are very, very vulnerable, which generally means that they carry out ecdysis in a hidden location so that they can stay there out of eyesight of predators until such times as the exoskeleton hardens up again. But on some occasions, predators, including ourselves, take advantage of this. So if you've ever eaten a Dungeness crab or a blue crab or a soft shell crab, you're basically eating the same crab. You're just eating them at different points in their life cycle. So a soft shell crab is simply one that has shed its old exoskeleton, but is still not hardened up its new exoskeleton. Hence the soft shell, which is easy to digest. And typically when you eat a soft shell crab, you eat the whole thing. Whereas if you get the crab with a hardened exoskeleton, it is tough to get through. Think about the last time you had a lobster or something like that, you had to use the special device that they provide you with to crack the exoskeleton so you can get the flesh out of it. It's particularly tough. And so you have to have ways of breaking that down in order to be able to get access to it. But even if you ingest a little bit of the chitin, no harm, no foul, because you can break it down. It is very digestible. All arthropods do have an open circulatory system. They have a hemocoel, not a coelom. And so the fluid is known as hemolymph, and it percolates through the tissues inside the body of the animal to move nutrients and to move gases around. In terms of gas exchange, there are a number of different mechanisms in play. If the animals are aquatic, then they have gills, and those gills can be located in a number of different locations, and there could be multiple sets of gills to allow them to maximize gas exchange. If they are very small, so that the surface area to volume ratio is particularly large, they may actually do gas exchange through the cuticle, which has thin regions that allow for gas exchange, and also for waste disposal simply by diffusion. So for very small arthropods, particularly very small aquatic arthropods, that's also a method for disposing of waste and also doing gas exchange. If the arthropods are terrestrial, then there are two different mechanisms. So the chelicerae, particularly the spiders and the scorpions, and I believe also the mites and ticks, use a modified gill apparatus called a book lung. So it looks a little bit like a gill. It is kept moist within the body of the animal, but it is exposed to the air, and it allows gas exchange through the thin layer of moisture on the book lung, so that oxygen can diffuse in and carbon dioxide can diffuse out through this thin layer of moisture, and then be taken up by the book lung. In the case of insects, they have a very novel way of doing this. It's called a tracheal system. These are multiple hollow tubes that penetrate into the body of the animal. They exit out through the exoskeleton through an opening called a spiracle. So there are multiple little holes in the exoskeleton. Behind each one is one of these trachea. That allows the gas to penetrate into the body of the animal. And because of the network of these trachea, the cells of the body are no more than 2 or 3 cells distance away from a tracheal tube. And so gas exchange can be by diffusion into the tracheal tubes. But this also limits the size to which insects can get. It's the reason why most insects today are so small. It's simply because the concentration of oxygen in the air right now, about 20%, limits the amount of oxygen that can actually penetrate deep into the tracheal tubes and therefore diffuse on into the tissue. And so to maximize the ability of oxygen to both penetrate the tracheal tubes and to diffuse into the tissue, the insects' bodies have to be necessarily very small, have a large surface area to volume ratio, and also position all of the cells of the body within a very, very short distance of the trachea. In the past, and we've talked about this before in a much earlier chapter, so you'd have to go back to the first plant chapter, which is chapter 29. And there we talked a little bit about the fact that during the Carboniferous, when we had the appearance of the first true forest, and we had a lot of plant growth being driven by conditions that favored ferns and horsetails and the like, oxygen levels in the atmosphere got much higher, upwards of 35%, we think. And this accounts for the reason why we have ancestors of our modern insects, very closely related, like giant dragonfly-like animals, giant millipedes, giant centipedes that are significantly larger than anything that we have today. So these are the dragonflies that have a wingspan of up to 3 feet. Centipedes and millipedes that could be 4, 5, 6 feet in length and maybe a foot wide across the body. This is the reason why they can get so big, because the oxygen concentration is much higher. That allows the oxygen to penetrate deeper into a larger body and therefore provide sufficient oxygen to maintain the integrity of the body. So these are all things that we know about arthropods, both from evolution and the fossil record, but also from our examination of modern insects. And this gives us some clues as to why insects and arthropods in general have been so successful over the course of their history on Earth. So when we look at the lineages of arthropods that are present today, we have the chelicerates, and this includes a very, very ancient arthropod that is referred to as a living fossil, and we'll talk about that in a couple of minutes. We have the myriapods, which are the centipedes and millipedes. Then we have crustaceans as a group, and then we have a group of organisms called the remipedians, which have gone backwards and forwards as being a sister group to insects or being part of crustaceans. Right now, I think they are mostly accepted as crustaceans, but you can see that the line here is a dotted line indicating that this is where we think they might be, but it's not definitive. And then we have insects, and notice that crustaceans and insects do form a common clade with a common ancestor. Hence the idea of the pan crustaceans, including the insects and the crustaceans in a single clade. So let's look at the cheliceraforms or the chelicerates first. So this is one of the most ancient clades within the modern phylogeny of arthropods. It includes the spiders, the ticks, the mites, the scorpions, which you're probably all familiar with, but then an organism called the horseshoe crab. These are marine, and they are the only marine examples of chelicerates that are extant today. So here's a picture of horseshoe crabs. These are endemic along the east coast here of the United States. They are particularly well known in the Northeast because at certain periods of time on a neap tide, which is the next to most extreme high tide, the horseshoe crabs come ashore at certain times of the year and have a mating frenzy on the seashore. The males and the females, well, particularly the males, vie for access to the females. You may have half a dozen or more male horseshoe crabs trying to mount a female horseshoe crab in order to fertilize the eggs that she is busily laying, while they secrete their sperm to fertilize the eggs as she's laying them in the sand. The reason why they lay their eggs at this point in time is because at the next high tide, which will coincide with the next full moon, the regions where they laid their eggs will be covered by the incoming tide, and the little juvenile horseshoe crabs will hatch out and swim out to sea on the retreating tide. Now, we do have them here in Florida. If you go to a region where there are mangroves along the seashore, you'll find little horseshoe crabs swimming in amongst the mangroves. I recall going over to Sanibel Island and walking along some of the nature trails there down in amongst the mangroves and seeing little horseshoe crabs swimming around in amongst the roots of the mangroves. At some time, on some occasions, the Orlando Science Center has had some horseshoe crabs in their mangrove display. They have a little artificial mangrove habitat, and on occasions they've had little horseshoe crabs swimming around in that model mangrove environment that they have set up inside the Science Center. So if you have access to the Science Center, you might be able to go and see horseshoe crabs in the Science Center. And again, these are living fossils. They have a long record in the fossil record, and they have remained essentially unchanged throughout all of that time, at least in terms of physical appearance. And amazingly enough, as weird as this may seem, people are now starting to look at horseshoe crabs as a delicacy. This is an idea that's emerged over the last 10 to 15 years. Some chefs up in the Northeast have figured out ways to prepare the horseshoe crab. And I guess just as once lobsters were considered to be trash and not worth consideration as for eating, and were for the very poor to eat, but are now considered to be delicacies and high dining. I guess that's what's going to happen to the horseshoe crab if the chefs have their way. Not sure whether I'd really want to eat a horseshoe crab, but then when you think about it, a lobster is just a seagoing cockroach. Yeah, no, that's a way of looking at it. And we eat lobsters. So there you have it. Anyway, onwards and upwards. So let's take these, you should be familiar with. Classic scorpion, the dust mite, very small, lives in your carpet, lives in your bedding, drifts around in the dust in your house, feeds on your skin, or more precisely, the little fragments of skin that you shed on an ongoing basis. That's their nutrition source. And some of you may unfortunately have allergies to dust mites. There's not a whole lot you can do about that, because no matter how much you vacuum your carpet, the dust mites are always going to be there. No matter how scrupulously clean you keep your residence, the dust mites are still going to be there. And you simply can't get rid of them. Then, of course, we have the spiders. Now, of course, spiders are well known for spinning webs, but what you may not be aware of is that only about 50% of all spider species spin the classic web. Many of them do spin other forms of insect trapping devices using their silk. So some simply secrete a single thread of silk with a bob of sticky material on the end, and they swing that backwards and forwards and try and stick that onto the back of a insect flying by, for instance. Others use burrows and nests that they've dug into the ground. They'll line it with their silk. They may even put a door over the end of it. These would be the trapdoor spiders and the funnel web spiders that come from my home country, Australia. Other spiders actually live underwater and are major predators in freshwater environments. These are the diving spiders. And they use their silk to create a little net that they can trap air bubbles under. And they will actually rise up to the surface of the water, stick their abdomen up in the air, and then slowly drag it down again, trapping air in the hairs on their abdomen. Then they swim back down to their little nest-like arrangement and deposit the air bubbles underneath the silk so that they create a little air cavity that they can breathe from by sticking their abdomen up into the bubbles of air. Then they can go out and trap things like tadpoles and small fish and things like that. And they're major predators on small animals in freshwater environments. There are all sorts of interesting things that we could talk about where spiders are concerned. Unfortunately, that would mean a whole new presentation just focusing on spiders and another whole new presentation focusing just on insects. And we simply don't have the time for that. So when we look at the basic makeup of a spider, you can see that there are multiple modifications of appendages. So the legs are obviously modified appendages. The chelicerae themselves, which are the venom injecting fangs, are modified, a pair of modified appendages. The venom itself is actually modified saliva that also contains digestive enzymes, which serves to digest the inside of the prey, an insect, for instance, and turn that into a soup. The spider then reinserts its mouth parts into the body of the animal and sucks that soup out, leaving behind an empty exoskeleton. And so the pedipalps and the mouth parts are actually, again, modified appendages. You can see here the book lung. This is the modified gill apparatus that is exposed to the air but is covered in a thin layer of fluid to allow gas exchange. That's connected to the open circulatory system. So the fluid is drawn in from the body, circulates around the book lung to pick up oxygen and to release carbon dioxide, then is pumped by the heart out through the vessel at the posterior end of the animal, and the hemolymph then percolates through the tissues towards the anterior end of the animal, where it is captured again and taken back into the open circulatory system. Spiders also, you're probably aware, have 8 eyes, and they can see exceptionally well with all of them. Not all of them are of equal quality. The 4 largest ones provide the greatest amount of information, but they also have some smaller primitive eyes that are basically for detecting the presence of light and shade. The 4 major eyes do provide clear images. We know that spiders can respond to images. They can hunt down their prey, so they're obviously able to see exceptionally well, and their neural system is able to process the input from all of these eyes to create what must be an interesting but very concise image of what is around the spider, because it is very, very difficult to sneak up on a spider. They see you coming a long way off. Also, interestingly enough, some of the larger spider species prey on animals that we might think would lie beyond the scope of a spider. So there are bird-eating spiders and there are bat-eating spiders. These have a venom that can paralyze their prey very, very quickly. Fortunately, they choose not to have anything to do with humans, but they are certainly significant predators on large vertebrates as well. Then we have the myriapods. The name tells you everything that you need to know about the body plan. Myria, many, poda, legs. So these are the millipedes and the centipedes. Millipedes do not have 1,000 legs, even though the name suggests that. Their proper name is diplopoda, and diplo, you may recognize, refers to 2, and this refers to the fact that they have 2 pairs of legs per segment along the body of the animal. Millipedes are vegetarian. They eat plant material, so they are harmless, unlike their centipede cousins. They also, along with centipedes, represent some of the first animals to colonize dry land, probably venturing out onto dry land even before plants colonized the land. And the reason why they could is because of the exoskeleton. It provides a watertight coating around the body of the animal, prevents them from dehydrating when in contact with air. And so based on the trackways that we have, it appears that millipedes, and almost certainly centipedes, perhaps even more so than millipedes, were likely the first animals to venture onto dry land simply because they could, and because they found opportunities to perhaps take shortcuts or to hunt down prey, in the case of centipedes, in ways that they may not be able to by remaining as aquatic animals. So this is a typical millipede. The largest millipede is actually the Madagascan hissing millipede. You can actually purchase those through specialty pet stores to keep as pets. They're very easy to look after. You put them in a little plastic tray, keep them fed with some decaying plant matter, and they're quite happy. And when you pull them out, they feel kind of ticklish walking all over your hand. I had an opportunity to handle one at the Orlando Science Center many years ago. And yes, they do make a hissing sound by drawing air in and out through the trachea in their body. That's where the hissing sound comes from. Then you have the chilopoda. These are the centipedes. They have a pair of specialized appendages associated with the jaws that are the poison claws that allow them to inject venom into their prey to paralyze them. Fortunately, the vast majority of modern centipedes are not considered to be dangerous to humans, although some of the larger centipedes in Africa do pose a known danger to small children, simply because of the amount of venom that they inject. For a human adult, it will be extremely painful. It could be potentially life-threatening, but typically is not. But for a small child, where the degree of envenomation is much greater, that is a clear risk to the child. So this is a typical centipede. We actually have a very tiny centipede here in Florida that lives in the grass. It's actually referred to as the grass centipede. These are typically about 1 inch long, and perhaps, about 1/10 of an inch across, so maybe 25 millimeters long and about 2 millimeters wide. They're quite small, but you do see them out running across the sidewalk from grass on one side to grass on the other side. I've seen lots of them running across at various times of the year. I guess it depends on how successfully they are in hunting on one side of the pavement versus the other side of the pavement. Then we come to the pan crustaceans. This is the big clade of arthropods. And it's just got bigger because it's now a merger of the crustaceans with the insects, or the crustaceans and the hexapoda. So let's take a look at the crustaceans first. So crustaceans are predominantly marine, but there are freshwater examples as well. So the crayfish that you saw dissected in the Invertebrates 2 lab is actually a freshwater crayfish. It's not a forgotten the term now. Anyway, it's a freshwater crayfish, and so there are a number of crayfish that are actually freshwater living found around the world. But the classic marine crustacean that most people are familiar with is either a lobster or a crab. They do have highly specialized appendages for feeding, for walking, for sensory purposes. They have the open circulatory system associated with all arthropods. Gas exchange is typically either via gills or through thin regions of the cuticle, or a combination of the two. Excretion of waste is also typically through the thin regions of the cuticle. Salt balance in those crustaceans that live in the ocean is either, again, via the hemolymph, or, as you saw in the lobster, a pair of glands, sometimes referred to as atennal glands or green glands or maxillary glands, that lie at the base of the large antennae. Remember that crustaceans have 2 pairs of antennae. That's one way that you can tell a crustacean from an insect. So at the base of the large antennae on crustaceans like the lobster, or the crayfish that was dissected in the video that you looked at for the Invertebrates 2 lab, you have this pair of glands sitting right underneath the base of those large antennae. Those are the glands that secrete salt and maintain salt balance in marine crustaceans. There are 2 separate genders. Again, you saw that with the crayfish. In the case of crayfish and lobsters and shrimp, what are members of the decapods, and we'll talk about that very shortly. There are usually tiny little leg-like appendages on the underside of the abdomen. These are referred to as swimmerets on the male. The most anterior pair of them are enlarged, and they are used for transferring a sperm bundle from the male to the receptacle of the female for the purpose of fertilizing the eggs. So that's one way that you can externally pick a male crayfish or lobster from a female. And that's kind of important if you're going to take advantage of your right as a Florida resident to take some Florida lobsters from the environment at a certain at certain periods of time during the year. You're only allowed to remove males. You cannot remove females, and they must be of a particular size before you can remove them. So if you're interested in doing something like that, you're a scuba diver or just someone who is interested in free diving to try and catch some Florida lobster at the right time of year, you need to read up on the requirements and make sure that you only harvest the lobsters that you are permitted by law to harvest. You're limited to taking 2, and they can only be males of a particular size. If you are caught with illegal lobsters, there's a huge fine associated with it. So let's look at the different types of crustaceans. So the first type is the isopods. These are typically small marine crustaceans, but if you've ever played with this, and you probably recognize it, it's a roly-poly, then you've actually played with a terrestrial crustacean. So the woodlouse, otherwise known as a pillbug or a sowbug, or in common terms, a roly-poly, is actually a crustacean, not an insect. It's a member of the isopods. Then we have the decapods. Obviously, deca is the prefix for 10, so 10 legs. They may not always be obvious. Let's take a look at the crab. So we've got 6 walking legs, 3 on each side, so that's 6. The modified claws on the front is another pair, that's now 8. And then what you can't see is a small pair on the back of the crab that represent the 9th and 10th leg. Then, of course, you can see the mouth parts here, again, highly modified appendages for moving food to the mouth parts and then the jaws themselves. And by the way, arthropod jaws do not move up and down like our jaws. They move like this, side to side. So it's a different jaw system altogether because it has a different origin. Then you've got the eyes on stalks, again, modified appendages. In this case, we don't have obvious antennae, but these extensions on the eye stalks actually serve as sensory organs for the purposes of substituting for the antennae. Then you have some very, very small crustaceans called copepods. These are things like the brine shrimps, and you can see the extreme number of legs, both on the thorax and the abdomen. So this is a reflection of the expression of the UBX gene as a promoter of legs, rather than as a suppressor of leg formation. And these are the animal portion of plankton, otherwise referred to as krill, that supports some of the largest animals on the planet. All the baleen whales, so the blue whales and the humpback whales and the like, and also the largest fish on the planet, the whale shark, because sharks are fish, and the basking shark, both of which feed on krill. They're filter feeders. Then there's one group of crustaceans that don't look anything like a typical crustacean. And the reason why is because the chitin shell is hardened with calcium carbonate extracted out of the water. So the shell actually feels more like a bivalve, an oyster shell or something like that. And in fact, the animals look very much like a bivalve. If they've retreated inside the shell, you might confuse them for a bivalve, but in fact, barnacles are crustaceans. And they have modified appendages to use as feeding tentacles. And you can see these barnacles have their feeding tentacles extended to filter food out of the environment. They are sessile. Depending on the type of barnacles, they may attach to a surface, or in the case of boring barnacles, they'll bore into a structure. And what is interesting is that if this is the structure that they're boring into, they create a tiny little pinhole that allows the larval stage to enter in. Then as the animal grows, it creates a large cavity inside the structure that it's bored into. So this could be the leg of a pier, it could be the hull of a wooden ship or something like that. They like, and these will burrow into concrete and all sorts of things. But what they do is trap themselves permanently inside, because once they get increase their size a little bit, they can no longer fit back out through the opening. So they have permanently trapped themselves in here. The only thing that they can extend out are the feeding tentacles. And they can be quite damaging to structures that they bore into, because they weaken the structure from the inside by creating these cavities inside them. So then we come to the insects. Previously known as subphylum Hexapoda, generally referred to just as insects at the moment, until it's determined exactly what to or where to place them within the biological hierarchy. When we look at the inside of an insect, and you should be familiar with this from the dissection of the grasshopper, and again in the Invertebrates 2 lab, you see the major organs. So it has a true digestive tract with a mouth, modified appendages to form the mouth parts, to move food in, and also to form the jaws. A complete digestive tract with the mouth opening, a cylinder to connect them, and then a second opening, the anus, at the posterior end of the animal. This animal is a female, so she has ovaries, but they are dioecious. There are males and females. The male has a sperm depositing organ that allows him to deposit sperm to fertilize the female's eggs. Often in a lot of female and to some degree male insects, but particularly female insects, you're going to find these yellow colored bodies. These are fat bodies. This is how the insects store extra energy, because typically if they are a migratory insect, and some species of grasshoppers known as locusts do migrate in large swarms from one location to another, and they're extremely destructive. They will store energy in the form of these fat bodies to allow them to move from one location to another. They are voracious eaters. They're incredibly hungry all the time. They're looking for anything that's green, and they'll eat anything that's green, not just plant material, but anything that's covered green, colored green, green paint, green cloth, anything that's green, they view as being a potential food source. And they're very, very destructive when they get moving. We used to have huge swarms of a grasshopper that lives in the Rocky Mountains called the Rocky Mountain grasshopper. It used to swarm out of the Rocky Mountains heading eastwards across the Great Plains. And they were extremely destructive during the late 1800s and early 1900s. We don't see much evidence of this swarming anymore because of habitat loss and the reduction in numbers. The swarming is actually a response to the fact that the adults and the hatching juveniles are in competition for the same resources. So in years where a very large number of juveniles hatch out and need food resources to complete their development, the adults, the existing adults, instead of remaining and competing with the juveniles, which would probably mean the death of the juveniles, they change form, and they physically change form from a benign feeding stage to a very aggressive migratory stage that is an aggressive eater. And this is the form that takes wing in these large migratory swarms. Just how big can a swarm get? Well, we don't really know. But we do know that the largest swarm ever on record in the United States took over 24 hours to pass any point that it flew over. And in terms of the size of the swarm, it's estimated that it extended from ground level to perhaps 1,000 feet into the air, and it was just solid grasshoppers. And the front of the swarm from one edge to the other was about 25 miles wide. This was a huge swarm of grasshoppers. And Laura Ingalls Wilder, the author of Little House on the Prairie, actually wrote about this swarm because it took flight when she and her family were living in a town on the Great Plains. It's written about in, I think it's the book called The Little Town on the Prairie. It was so destructive that Laura's father actually had to leave town and walk about 400 miles eastwards to find work in order to be able to earn some money to send back to the family so they could support themselves until the next spring, and they could start planting crops again to support themselves again. It was extremely devastating. So flight is one of the adaptations that actually accounts for the overall success of insects. The vast majority of insects actually have wings. Whether they use them to fly or not is not always the case, but the vast majority of insects do certainly possess wings. They may just not necessarily use them for flight. Most insects do have two pairs of wings. Some use both pairs for flight. In the case of grasshoppers, as you discovered, one pair of wings is actually the flight wings. The other pair of wings is used as a protective cover over the flight wings so that they don't get damaged when the insect is not flying. The covering wings do contribute a little bit to flight, but they are not the primary flight wings. Insects like butterflies and moths do have two pairs of wings that overlap and appear to work as a single wing, but in fact there are two pairs of wings. And then flies, like our model genetics organism, the Drosophila, have a single pair of wings. They do have a pair of stumps, I guess you could call them, called halteres, that are posterior to the existing pair of wings on the thorax. They would develop into a second pair of wings, but that wings development is suppressed. But there is a mutation in Drosophila that actually promotes the growth of that second pair of wings. So this is another one of the many mutations that geneticists have identified in fruit flies that we can track from generation to generation. Exactly where wings came from is not entirely clear. There are two hypotheses for the origin of wings. One is that because of the venation that most insect wings possess, it's possible that a smaller version of the wing served as a heat dissipating or heat absorbing device that expanded over time to release or gather more heat, but and of course to if you're trying to remove heat, moving the wing up and down through the air or moving the device up and down through the air would allow for better dispersal of the heat energy. If it gets big enough that moving it up and down then allows for the insect to take flight, that would explain the origin of wings as something that has been modified for a new use simply by expanding on its existing use until it takes on a new function as a consequence of modification. The alternative is that the wings may have served as some form of device within fresh water, because some of the earliest flying insects, the dragonflies and mayflies, begin their life as aquatic nymphs, their larval stage. And they only get their flight wings when they go through the last period of ecdysis that precedes the emergence of the adult. That's when the wings emerge. The wings are not present on the juveniles, they are only present in the final adult stage, although you can see structures that are clearly going to become the wings developing during the last few ecdysis stages called instars. I mean, every time insects that do not have or do not go through complete metamorphosis, and I'll explain the difference shortly, although we've encountered this before, as they go through the sequence of ecdysis steps, that results in them becoming more and more adult, we refer to each of those stages as an instar. So they go through multiple instars, and it's only in the last few instars that you start to see the emergence of the structures that will eventually become the wings, only emerging as fully mature flight wings in that last ecdysis step that results in the emergence of the adult. So you have these two hypotheses. Obviously, they're mutually exclusive. You have either as a heat related device or as a device in water. It's possible that flight may have emerged more than once in insects, which might allow for both hypotheses to be correct. We don't know for sure, but all we do know is the vast majority of insects do have wings, and it accounts for a lot of their success. It's one of 3 ideas that accounts for their success. So we've talked a bit about the different types of wings. We do know that insects were emerging during the Cretaceous, and certainly became highly diversified as a consequence of having to deal with the resinous material that gymnosperms secrete in order to allow them to feed on gymnosperms. But that diversity was greatly reduced during the Cretaceous extinction, or the end Cretaceous extinction. That's the one that took out the dinosaurs and other species. But subsequent to that, we've seen an increasing number of adaptive radiations of insects that has increased the diversity of insects and the sheer number of insects. And a number of those either have been as a consequence of the adaptive radiation of flowering plants, or flowering plants diversified as a consequence of the emerging diversity of insects. The hypotheses have gone backwards and forwards between the 2 ideas. The current thinking is that it was insects first, leading to diversity of flowering plants. That may change in the future, but that's the current thinking. So we do know that insects undergo metamorphosis. There are 2 types of metamorphosis, and we've talked about this before with respect to other animals, but in insects, the idea of metamorphosis is the one that we most associate with. So incomplete metamorphosis is where you go from a juvenile that is somewhat like the adult through a series of stages or molts or ecdysis steps called instars, as I said. And with each successive molt, so this is where the grasshoppers get into trouble during years where there are a wealth of resources for them to eat, which gives them the opportunity to reproduce in huge numbers. The emerging juveniles, as I pointed out just a few minutes ago, now have to compete with the adults for access to resources. The adults change into the migrating form and start migrating, looking for resources to tap into, leaving the juveniles behind to utilize the resources that are there. Doesn't mean that the juveniles are necessarily going to be successful because of their large numbers. They may actually eat themselves into oblivion because they exhaust their resources and only a few will survive, which means the numbers left behind to reproduce will be much smaller, which means the next year, smaller numbers will emerge, which is the reason why you don't see a swarm every year. The swarms come in a cyclical fashion. The unfortunate thing is that because of the advent of modern agriculture and the tradition of growing crops, particularly monoculture crops to support humans, this has increased in some locations, particularly in the poorer regions of Eastern and Northern Africa, the frequency of the swarming of the locusts. And it really has put people at risk, because every time the locusts go into swarm, they eat all the crops before they can be harvested, which leaves the people without any food and creates famine conditions. So these are the sorts of things that are associated with incomplete metamorphosis. The juveniles will always be in competition with the adults for access to resources. Other insects undergo a complete metamorphosis. So this is the sort of metamorphosis that we associate with the moths and the butterflies. In the pupal stage, in this stage, the larval body form is completely destroyed. Leaves behind a soupy residue. So in order to create a brand new body plan in terms of the adult, the moth or the butterfly, you have to completely disassemble the larval body plan, the caterpillar. And this is what takes place inside the pupa. The material that's left behind, as I've indicated in the note, is a soupy material. It contains lots of cells, and those cells then reorganize themselves, start dividing, and start generating a completely new body plan, which is the body plan of what will become the moth or the butterfly. It's a truly amazing process. But in order to get the butterfly, you have to completely disassemble the caterpillar, completely disassemble the larval stage in order to generate the adult stage. So they are dioecious. There are males and females. The females, let's see if I can fit this in here. So, and this is particularly true for moths and butterflies, but the females typically produce chemicals known as pheromones. These are sexual attractants. They are released into the air. The males can sense these using their antennae, and they follow the trail of the pheromones to the females. That's how they can identify. That's how the males can identify that the females are in the area and that the females are available for mating. You may also know that some insects, along with some spiders, also practice carnivorous behavior. during mating. That is, the females, because they are about to carry the burden of raising young, need an energy resource, and the male is sitting right there, and he becomes their first meal as a pregnant female. This is the case in mantids, the praying mantises, and also many spider species do this. And in the case of the spider species, yes, I realize they're not insects, but they have this issue as well. It almost seems like the males are aware of their fate, because a lot of them will actually capture an insect, wrap it up in silk, and carry it in their most anterior pair of walking legs and hold it in front of them as if to present this to the female and to say, here, look, I've got something good for you to eat. Leave me alone. Please don't eat me. Please don't eat me. She still eats them because she's going to be, as a pregnant female, very hungry and looking for lots of food to support her ability to support the young during their very earliest development, and before they hatch out. She's got to support the eggs and support the brood. These are but a limited number of the various clades that are present within insects. So here you can see a number of those that are capable of flight, and some that are not capable of flight. But Orthoptera is, actually all of these are capable of flight now that I look at it. So Hemiptera are beetles, or beetle relatives, I should say. And they do have the ability to fly, although they may not fly very well. Orthoptera are the grasshoppers and the crickets. Coleoptera are the true beetles. Diptera are the flies. Hymenoptera are the wasps and the bees. But we also include in there ants and termites, because they are closely related. And then Lepidoptera are the moths and the butterflies. So that brings us to the end of the arthropods and to the end of the ecdysozoans. We have one more major clade to look at, the Deuterostomia. But before we go there, let's take a short break and consider these concept check questions when we come back from the break. Explain how the nematode and annelid worm body plans differ. I left out the in there, unfortunately. Outline 2 adaptations that have allowed insects to be so successful in terrestrial habitats. And I have actually identified 3. So when we come back from the break, I'll answer these questions. See you on the other side with the answers.